we've had several good discussions already on epigenetic clocks and DNA methylation, so I don't need to go through the background. I have the benefit of going after others to explain clock so well. And so I'm going to talk about a novel clock. It's called Physiological Health Age, or PhysAge for short. That's designed with several key advantages over prior clocks, and one of them is the interpretability of this clock for potential monitoring. And so I'll put out there that we're all in agreement, though, that this isn't ready for, as Coulter said, prime time. It's more for group level, population level work and monitoring potentially within individuals. So we're not advocating that this is going to be sold and really. So for those not as familiar with just the idea of the clocks or development, I think it's worth mentioning what we're trying to find with these clocks. First, the answer rests on the premise that chronological age is the biggest risk factor for declining health, disease, and mortality. And what we want to find with these clocks are a proxy measure for age that predicts health and disease and mortality better than chronological age. So our interest was in to initially develop an epigenetic proxy that would reflect the state of physiological health. And that epigenetic proxy or clock would need to reflect a degree of accumulated damage and unmeasured exposures throughout a person's lifetime. So we decided to train the epigenetic surrogates on biomarkers of health that, when put together into an index, would yield a proxy measure. So identifying these biomarkers had already been completed by McCrory and colleagues in a 2023 meta-analysis. And so in this meta-analysis, they took individual participant data, conducted this meta-analysis on about 67,000 people, aged 40 to 110 in 13 cohorts. And they developed a multi-system index to reflect the state of physiological health. And then they tested independent associations of 40 different biomarkers across 12 physiological systems for associations with health outcomes. So they specifically identified a panel of 8 biomarkers representing different physiological systems that were reliably and consistently associated with health outcomes, including walking speed, grip strength, and self-reported health. So the biomarkers identified included HDL, cystatin C, waist to height ratio, glycated hemoglobin, CRP, heart rate, peak expiratory flow, and dehydroepiandrosterone. It's harder to say, I guess. So they also found that when they created a composite score from these 8 biomarkers, when they created a composite index of these 8 biomarkers, it strongly predicted mortality and represented a measure of multi-system dysregulation. So we built off those findings to create epigenetic proxies or surrogates for each of the 8 biomarkers. So first we needed to prepare the data for this that we started with, and we started with HRS, the venous blood sample. Participants who had complete data on the biomarkers and DNA methylation, which was 3,177 people. We split them in half into a training and test set. You can see they're about 68 years old, diversity in sex and gender, and race and ethnicity. So then we trained individual surrogates on these 8 biomarkers. Although instead of heart rate, we used a measure of pulse pressure, which was diastolic subtracted from systolic blood pressure. And then we took the 8 biomarkers, and the training sample used elastic net regularization to develop an algorithm, and then use the algorithm to identify the CPGs that predicted each biomarker separately. So the identified CPGs and respective beta coefficients from the regularized prediction models were then used to calculate each surrogate in the test sample. So each surrogate was calculated as a sum of the weighted values for each CPG and then standardized. Then we also summed the standardized surrogates into the single score, which represents the epigenetic proxy, the physiological health age score. So to validate the surrogates, we did several things. This is just to show that we compared the surrogates against their clinical biomarkers in all three data sets. That middle one is tilde. I don't know why that doesn't show. There it is. Oh. So I didn't squish these in to accentuate the lines or anything. I just had to fit it all on one slide. So it's all there, and you can see that the correlations ranged between 0.28 to 6.4, depending on the biomarker. But you can see that the correlations basically recapitulated across the validation studies. So then we compared how the surrogates performed in predicting health outcomes, including gait speed, grip strength, cognitive errors, and frailty. So peak flow seemed to be the strongest predictor. DHES was probably the weakest, but you can see a pattern of prediction here that was relatively strong for each of the surrogates. The scales for the surrogates of HDL, DHES, and peak flow are reversed so that all the surrogates are expected to show the same direction of effect here. And with these models, we adjusted for age, sex, race, ethnicity, white blood cell counts, and then associations for gait speed and grip strength were also adjusted for height. We also evaluated the predicted performance for functional ability, the ADLs and IADLs, subjective self-rated health, and 6-year mortality. So these are in the HRS only, but we saw similar patterns with tilde and Nicola. And then we also compared how well the surrogates performed against the clinical versions of the biomarkers in predicting these outcomes. So here, the bottom row is Nicola. We see the three cohorts for these three outcomes: gait speed, grip strength, and cognitive errors. And with each biomarker, each one is depicted by a different color. But when the surrogate performs better than the clinical biomarker, you'll see a diamond. When the clinical variable performs better, you'll see a filled in circle. And then when it's the same, you see a olive circle. So what this shows for many of the measures is that the proxies are capturing additional information to predict health state that the clinical biomarkers may not be. They seem to be predicting better in almost half the cases, or half the biomarkers. We see a similar set of plots there for frailty, ADLs, and IADLs in the three samples. And again, we see at least half of the surrogates are performing better than the clinical measures in predicting each of these outcomes. And then we also looked at self-rated health and mortality. And we can clearly see the strength of the surrogates with more diamonds and circles predicting mortality. This was 6-year mortality for HRS. It was about 12 years in tilde and about 11 years in Nicola. So next we evaluated the performance of the composite PhysAge score. And here we see the correlations with the other second generation clocks. And now I'm going to call these conspiracy plots because they're but essentially we just see the correlation with a PhysAge with PhenoAge at 0.57, 0.61 with GrimAge 2, and 0.65 with Dunedin pace. So then we compared the performance of PhysAge to its clinical composite score, and you'll see that at the top row, the PhysRS, that's the clinical score, and how that performed in predicting the same health outcomes that we've been looking at. PhysAge is highlighted in the blue rows, and we compared it to Pheno, Grimm, and Pace. And you can see that it performs as well, or in some cases better than the other clocks. And we did this in the 3 cohorts, so this is HRS. And then we saw similar patterns of performance in tilde on the same health outcomes. We see similar patterns of performance in Nicola and the same health outcomes also. And I'm going through this quickly because I want to get to the interpretability part. So this is essentially showing that we're validating. Oops. And then I wanted to highlight one more thing, is that the prediction of mortality in particular, PhysAge is depicted by the blue lines in these charts, but it's difficult to distinguish from the other lines because the performance is nearly the same as Grimm H2 and as PACE, and it's slightly better than PhenoAge when you calculate AUC. So how can we make more sense of this clock in particular and extract more intuitively useful information? So we propose to go back to the constituent surrogates of PhysAge and use them to unpack what the proxy is reflecting with multi-system health. So to demonstrate this utility, we transformed the surrogates into age metrics of years. And so this table shows PhysAge and its component surrogates, all expressed in years, for 4 people, all men, who had the same estimated PhysAge at about 50 years. The values displayed in green depict where the DNA M age is less than chronological age, and the red values indicate where the surrogate exceeds chronological age. So for example, in person 1, who has a calendar age of about 64, but an estimated PhysAge of 50, the only biomarker on which they show age acceleration is HDL cholesterol. And so that seems to be driving driving some change. And for example, person 2, by comparison, has a calendar age of 59.6 and a PhysAge of 50. And so they display an age deceleration or an age gap for 4 biomarkers and an age gap for 4 other biomarkers. And so this likely reflects some aspects of their health that require closer monitoring and helps us to understand what's driving the age difference. Yeah. So here's another example of 4 women with an estimated PhysAge of 80. So person 4 in this table has a calendar age of 60, but a PhysAge of 80, and shows a striking age acceleration across all the surrogates except for DHES. So these values might identify that the person is at risk of poor outcomes and suggest where there are potential areas for improvement. In contrast to person 3 that shows just one measure, pulse pressure, that's potentially driving an age gap. And it suggests monitoring.
How did you get, maybe I missed it, how did you get from the surrogates of each of these biomarkers to ages, to years?
So yeah, so when we created this score for each surrogate, when we calculated a sum of the weighted values, we then took the standard deviation and the age of the training sample and used that to transform that score into an age. And so it was in the same age metric as PhysAge.
So does it predict age directly, or you would always need chronological age, the predicted population?
Across all of these cohorts, we just used the same age and standard deviation from the training sample.
Okay.
So across all the cohorts, basically the mean age is the same.
And sorry, and you didn't say how you combined them. You said they're weighted average.
Ah, sorry, that was, so we summed them.
You
So,
summed them.
so after we had the standardized score, we just summed.
Yeah.
So overall, this sort of shows proof of concept for the potential of using surrogates to stratify individuals based on risk projections for potential monitoring, still in group, group settings, not necessarily for individual at this point, and that may provide a way of unpacking what's happening with a single age, biological age score. So overall, PhysAge seems like it could be a useful tool for profiling differences in biological aging. The surrogate markers of the established clinical biomarkers predict physiological health states, potentially better than the clinical biomarkers. The surrogate markers are potentially better. And the surrogates have potential utility for group level trials where they can be used to assess changes in pharmacologic or other therapeutic interventions. So we think so in future directions, evaluating the temporal shifts in the epigenetic changes or particular CPG sites that contribute to the individual differences would be useful. And then testing in younger samples, including those studies where some of the biomarkers are not routinely assessed, which could make assessing this an advantage. And also the idea of putting multiple clocks together for greater interpretability, metabolic, transcriptomic components, and disentangling what's driving the associations with health outcomes. So there are many clock developers in the room. Many people have done some QC and validity, so it's a welcoming discussion on this idea of interpretability. Oh,
I have a question. So I understand the you have split it into its individual components. Are you thinking that if we develop interventions targeted to specific components, that it would actually change your overall surrogate score? Is that how you're thinking about these individual components? Like, somebody is, I don't know, higher on HbA1c, or another person is higher on CRP, so you would give targeted intervention to those specific biomarkers, and that would change the surrogate overall?
the idea that that specific component may be driving that difference in PhysAge versus chronological age, I think.
Yeah, I mean that's basically the concept. So you can see we were challenged, and it's a fair question. The reviewer said there's other clocks out there, GrimAge and Dunedin Pace, or the natural comparators amongst others potentially, and they're like, so what does your clock give that the others don't, right? So GrimAge again is built of inflammatory proteins, right? So Argulates reads out, it provides a readout of inflammation burden, plus other things like smoking pack years. It's really deleterious for your health. Dunedin Pace is a rate clock, but it's 19 biomarkers over that 20-year span distilled into methylation at a singular time point. So we were thinking in terms of what this one does is you can deconstruct it back into its component constructs and arguably then use it for, up until we saw some of the stuff earlier today. For exactly that type of thing. So it might be more informative for someone if you know that your biological age is accelerated. You might go, okay, look, seem to be doing well on these ones, but I'm performing poorly on that particular biomarker.
Yeah, I mean, we've thought about this idea, but we are afraid if we change the individual components, we'll just break the clock.
What the clock?
Break the clock.
We'll break the clock. That so I guess I'm thinking about there is this connection between the clock and mortality. or some adverse health outcomes later. To provide an intervention, I don't know whether you're going to break the connection between the clock and mortality or the adverse health outcome in the future, or what it is that we do. I mean, it's very interesting. I really enjoyed it, but that's the piece that I'm not sure if we intervene on the individual components of the clock. What are we actually doing?
But I guess
But-
at least you'd have a start of where to intervene. I mean, what do you think, what would you do about PhysAge or?
Oh, no, I don't have a solution. I'm just saying.
Case, I mean,
Yeah.
where would you start?
I mean, this was like related to the question I asked yesterday, like, if you were to monitor biomarkers and any kind of aging intervention, how do you actually
Yeah.
monitor the efficacy of that? And if you're using a surrogate marker, and this is one way of thinking about it.
Yeah, we were thinking this in the context, so the kind of simple one is if you start to take a statin, what it does to your HDL level. If you start taking metformin, what it does to your HbA1c or something like this, right? So potential uses of that. But on the other side of it too, we're thinking, so the individual DNA methylation circuits themselves have utility, as we know from GrimAge, is kind of first proof of concept. But what I think of GrimAge is a beautiful clock, but it's like, what do you do for someone who's got a high TIMP1 or PAI? What does that mean for someone, right? Whereas pulse pressure, HDL cholesterol, HbA1c have an empirical reference. They're clinically useful as part of the natural screening and profiling. So as I said, we think it as a proof of concept at the moment, but I do think potentially has utility.
Can I just ask, so actually purely not biological or medical. The thing I thought was interesting is taking those same things and somehow quantifying, like, is it one biomarker that's really far out? Or is it several, you know what I mean? Like, that's actually like a derivative measure that I think would actually be pretty interesting, right? Is this driven by one or two, or is it driven by all of them a little bit? I think that there's something potentially interesting there. Has no clinical relevance directly, but it's like a secondary measure.
Yeah, because it more closely tracks those biomarkers. Because I mean, I guess I didn't explicitly say that this is also different because we didn't train on age or mortality.
Right. No, and I don't know, it feels like that could be something, maybe you've already done that, but
Do you think if you did train on mortality, you would outperform PhysAge? Substantially.
I think we could have done a third step of selection here. So one way to trim this was to elastic net those biomarkers, or another way to do it was to drop the HEAS. It's the obvious one, didn't perform very well. So, but we were kind of committed to having that remain faithful to the original composite score that came from the 13 cohort studies as opposed to one.
Okay, thanks for your patience for me getting my slides up and running. Appreciate it. So my talk today is going to be interplay between omics and outcome, and I'm Rob Leigh from the Tilda project in Trinity College. I'm their resident bioinformatician. So prepared to be for. Oh, it's not working now. Is there anybody computer? Okay, so just the contents of my talk today, just an introduction to omics, exactly what I'll be talking about. What I'll be talking about, while other people talked about epigenetic clocks and stuff like that today, I'm more going into the gene biology. So I'm just going to give a few little graphs, kind of show what I'm talking about, why it's important. Kind of conceptualize this strange life thing that we all seem to inhabit. So then I'm going to move on to omics and outcome, how they interplay. And osteoporosis, which is the outcome that I've chosen to pick. When I chose the title for this talk, I wasn't exactly sure how my results were going to look, so I kept it vague. Then I have a differential methylation analysis in osteoporosis, which was very nice. And then I'm going to present PhysAge, a bone density predictor and risk estimator. So, an introduction to omics. So this very complicated and horrible looking slide just starts off by, if we look at the left-hand side, it goes through the 5 pillars of omics. So from start to finish, it's genomics, epigenomics, transcriptomics, proteomics, and metabolomics, of which I'll talk about the first 3. So within each of our cells, the entirety of our life, the entirety of our sequence is made up by 4 base pairs. A, T, G, and Z. And what we're interested in as epigeneticists is C and G, how they come together, and then the little methylation site that binds them that prevents or either starts a gene in its transcription process or prevents it from being transcribed. So turning a gene into a messenger. So just moving on from there. And then another thing that I'm interested in particular when it comes to these gene biology is the chromosomes, where these CPG sites sit and how they interact with the cell as a whole and the outcome then therein. So just the basics then, chromosomes, they store the DNA all together on these graphs here. The white bands are low density regions, the dark bands are dense regions. Light bands have more genes, much more genes. They're much easier to transcribe, and that's where most of the CPG sites sit. Okay, then just kind of start from here. The genome is the complete set of DNA, genes, and junk in an organism. It's the circuit board of the process. Every cell in your body has the same genome, but what makes a liver cell and a brain cell different is the epigenome. And these are the modifications that sit on top of the gene sequence without altering it that allow genes to be turned on and off to do different functions. And then the transcriptome, that is the message that comes from the CPG sites that allows for proteins to be constructed. And then for the rest of today, I will talk about the methylome, and we're going to move into more interesting things now. So the reason why we're interested in the methylome is it's a stable response to the environment. Unlike the microbiome or things like that, it's much easier to capture changes over time without much difficulty. In the genome, you see very stable responses, but it's mutations and they're not very informative in every case. The epigenome is. So again, it's a chemical, not sequence-based control switch, and it can be inherited and influenced by the environment. So there are inherited patterns here as well. It all depends on the state of the mother and the father's sperm and egg cell, the conception of life. And it's dispersed throughout the genome. So it's not just concentrated in one area. Now, when it comes to any individual cell, we see a bimodal distribution like this of CPG sites. Those on the left, the unmethylated sites, they allow for genes to be expressed, and on the right, as methylated, they stop genes from being expressed. And this is what we see whenever we profile any cell, and getting the balance of these in any study is kind of paramount to understanding how a clock works or how any cell or any system survives. And it's usually when this shape starts to break down, that's when you start to see disease processes. So each cell has that. Chromosomes may be differentially methylated, so you can see a lot of things moving from unmethylated to semi-methylated, for example. And then if we sequence it across a gene and we look at the CPG sites as they appear in a gene, not on a chromosome, we do see strong biases. We see that CPG sites are more likely to start the gene, i.e. they turn the entire gene on or off. If they're found in the center of the gene, such as the first intron or the internal intron, that means that they will turn a transcript into a different protein. So you see differential proteins coming out here. Now, and then this is just a schematic kind of showing what I'm talking about. There's a thing called RNA polymerase that binds to the DNA, transcribes it to RNA, and away it goes. So what happens is when we get methylation, that blocks the RNA polymerase from binding, and then it cannot transcribe its gene or make its product. And in any cell, let's just say we have our 3 CPG sites here. The difference between the cancer one and the others that aren't cancer is 1 CPG site. And in this case, it's a tumor suppressor factor that has been turned off, and that cell is now technically a cancer cell. That'd be the main difference. Now, omics and outcome. So, so all of these factors here that are associated with aging can be measured in some form using omics technology, and has been done so clinically and in the research environment. So we're already doing this in practice. So anything new or anything advancing on this is always good. Also keeps me in a job. So let's go with that. So microbiome modulation, for example, is reported in several diseases, multi-species, multi-system, and it's environmentally responsive. And then the inflammasome, allergome, and epigenome and mitogenomics all show considerable promise, especially in terms of accelerated aging. And epigenomics is the most stable environmentally dynamic measure. So osteoporosis. Now, osteoporosis is quite a big thing in Ireland. This is why we kind of chose it as our outcome. We also measure T-score in tilde, and it just seemed like a nice one. So I did that one. So just to kind of talk you through this now, the T-score correlates strongly with porosity. The more porous the bone, the less dense it is, the more likely it is to break. So the T-score, if it's greater than or equal to -1, it's normal density. If it's between -1 and -2.5 is osteopenic, which means that the pores are now widening. And if it's less than -2.5, it's osteoporotic, which means that it's now more likely to break. So why is this important? Well, it affects 200 million people. It's sex disparate, 33% of women versus 20% of men over 50 will have fractures. And sex disparity is likely due to calcium metabolism in pregnancy, where calcium is leached out of the bone by the child and donated by the mother. There's a significant mortality in frail populations. There's a 24% chance of hip fracture patients dying within a year of their fall. And significant potential loss of independence. 50% of survivors lose mobility permanently. When I say survivors, I mean hip fracture survivors. And a fracture almost doubles the future risk of more breaks. Hip fractures to rise to 310% and 240% of women by 2050. So that's compared to today. The economic burden is about 19 billion in US dollars. Prevention is better than a cure. Calcium, vitamin D, exercise, the usual suspects. And less than 20% of fracture patients get proper treatment. So the most prevalent reason for nursing home admissions in Ireland is falling with breaks and then a loss of independence. So in Ireland, what I did to produce this graph, I just took our T-scores and I just pretended they were weather stations and I plotted them out using a grading algorithm. But as we see here, it worked quite nice. As we see here, I don't know if you can see that mouse, can you? You can. So this follows a nice mountain range. So for whatever reason, your T-score is higher if you live higher. Don't know why that is, but it was nice to see. The reason why I made this, I wanted to see if there was areas of unmet need. And we do see that with the female population there. I couldn't even scale this to be on the same scales because we ended up losing too much information. So the females have much lower T-scores than the males do. They seem to skew it completely. So then differential methylation and osteoporosis. So what I did was just a normal standard differential methylation analysis, except I then took the statistical measures afterwards, and I ran them through a Gaussian mixture model to get this trimodal distribution. And anything in orange I took as not to be noise. I took that to be statistically significant. And then we got approximately 10,000 sites remaining. Nearly all of them were on the sex chromosomes. So we did not expect to see this. This is why I'm presenting this today. So most of them are on the sex chromosomes, and this is after we control for sex. So all sites, yeah, most are on the sex chromosome. If we move over here to the right, I just took one site per gene. If anything was augmented on any gene, I just calculated up to the far right there, and you see that pretty much every gene, it's only 1 or 2 per chromosome, but nearly every gene on the X and Y chromosome are affected in osteoporosis. And we did not expect to see this. And then if we plot that out in a nice, pretty Python package that I found, we can see that it seems to be agnostic to density. Usually you'd expect to see more CpG sites methylated in the white regions on the bottom, but they're also methylating the darker regions where you normally wouldn't expect to see them methylated. So the entire chromosome seems to be affected. Now, when I went and I tested what these genes were and their interactions, I got this lovely blob here, which just means that all the genes are interacting with each other in a big kind of complex, so doing lots of different things together. So what we found was that the associated genes are actually usually only studied in children, where they're usually prominent in X-linked syndromes. But when I went and I digged into the research properly, I found that a lot of the associated phenotypes, even though they were in children with disabilities, seemed to look like frailty measures or symptoms of frailty. So tremor, impaired confidence, hearing impairment, cognitive impairment, decreased body weight, and muscle weakness were the ones that popped out most frequently. And then allosome replication may explain some of the results. So the allosome is a sex chromosome. Men only have 1 of each. Therefore, when their cells are undergoing mitosis and meiosis, they can't undergo homologous recombination and repair their DNA properly. Therefore, any way or any mutation or any epigenetic buildup of plaques or whatever, they propagate the whole way down. They don't recombine and get shifted off. So there is much less repair mechanism in the male sex chromosome. So yeah, so as well. This may offer some of the explanation of differential aging. Osteoporosis is already sex disparate. Well, maybe it's a window into some of the other conditions we see also. Now, the X chromosome is significantly enriched for neural genes. Now, that's not just cognition, that's signaling as well. So that might explain some of the neurological differences we see in aging as well. It could be a sex chromosome breakdown. Now, when I ran this against all of the clocks to see if I could use that to predict the T-score or the osteoporotic rating, I couldn't. I ran them on every clock that was available in BioLearn. Couldn't get any of them to work, or any of them to be significant. Sorry, I should say none of them were significant. It's not I was trying to make them significant. So very YC CI distribution, all of them had long tails. Likely that the osteoporosis CPGs and surrogates are overlooked, kicked out by the elastic net algorithm, or just weak in general. So surprising as some of the surrogates strongly correlate with all of the other frailty measures, especially the Mateo data set. So I'm not sure why this one isn't working. So then I thought that, okay, well, look, if the osteoporosis is there, chances are there's pores, cells mightn't like that, cells come from the bone marrow, maybe they'll help me. So I estimated all of the cell types, thinking full well that the cells would tell me something, and they did not. So very surprised at that, and to be honest, I'm still a bit upset about it. I was hoping for a nice easy win, but I didn't get it. It doesn't affect differentiation whatsoever, apparently. So then, because I can't take no for an answer, I developed PhysAge, which is a bone health predictor. So it does risk scores as well, but I'm perfecting that part at the moment, so I'm not going to present the risk score today, but I'm going to show you its prediction power. So PhysAge, there is no prominent epigenetic predictor of bone health that I found. There probably is about 10 of them, but I didn't see them. Machine learning and classic statistical influenced feature selection is how I chose it. So I was using the BBMM decomposition to break the distributions of the statistics into the three Gaussian distributions to reduce noise. And then I got that. And anything in blue I took as non-noisy data. The boundary here is where something, for example here, is where something is more likely to be in the blue distribution than the green distribution, so completely out of the noise range. And the way I chose these were FDR corrected Spearman rule correlations. FDR corrected for the p-value, and then I modeled the rows like that. So they're cross-chip compatible. I restricted to CPG sites common to both methylation sequencing arrays, and they had to be involved in a gene so I could actually figure out what was going on. Otherwise, it's not fun. And then I ran an elastic net regression. I also did random forest. Random forest was really good for classifying, but not for T-score. So grid search then was used to hypertune parameters. I'd sanitize the data using inlier conversions with robust Z score. So what that means is I used a robust Z score to find any outliers, and then I imputed that with by taking the mean of the outlier and the closest inlier and repeating it until no more outliers existed. And I got this sanitized data set that I was able to build a model for. So then I ran it out, and then I had to rescale my T-score because in order to run this, I had to scale the T-score into one direction. That's why there's two measures here, and we see that the green is the measured value, the orange is the scale of predicted value, and the blue is the predicted. So the orange value is the one that we're interested in. It's quite close to the green, which I was quite happy with. But I'm not as happy until I saw that. When I got a R of 0.97 and a rho of 0.967, and my test data fell where it should. So the model doesn't seem to be overfit. Seems like it worked. So I went back and I applied it to the tilde data and like the non-sanitized version of the tilde data, and I got an R of 0.951 and a rho of 0.952, which I thought was pretty good. So then I wanted to see if it could classify stuff because T-score, you know, at -1, it's healthy. Between -1 and -2.5 is osteopenic, and less than -2.5, it's osteoporosis. So I just did some classification tests on it, like the confusion matrix. And the worst score it gets is 77% correct for osteopenia. And it's 80% accurate for osteoporosis and 91% accurate for normal bone as a first round hash-like. And any of the errors that we don't get are due to the closeness of the boundaries. So we see that the errors here are where we would expect something to shift into osteoporosis or osteopenia. They're kind of falling maybe one or two little spaces between the two. There's very, very little difference between them, but it's the classification boundary that puts them into the wrong one. So summary, osteoporosis measures such as T-score is strongly allosomal, so it's sex-linked. Almost all significant sites are X or Y linked. Existing surrogates do not seem to capture osteoporosis. Cell compositions do not predict osteoporosis. But PhysAge can reliably estimate T-score. Now, it does need more validation, so if anybody has any CPG sites and T-scores, please help me. Early work is promising so far, and more work is needed to determine sex differences in models. Many studies control the effect of sex, and I think we're losing signals like this by doing so. So I want to thank Professor Dr. Cathal McCrory and Regis Roseanne Kenny for their supervision during this project. Bill is supported by the Department of Health, HRB, Atlantic Philanthropies, and Research Ireland, and all of these guys. So, any questions?
It's a very exciting talk. I wonder, so you did pre-selection of features
Yeah.
before running machine learning, right?
Yep.
Did you use the whole data set for that or only training set?
Did I, sorry?
Did you use the whole data set for feature selection or only training set?
So the first round of feature selection, I did my differential methylation. I took what was significant there, then ran correlations, and I took that as the feature selection set. Then I split it into test and train.
Then you may have some data leakage, because if you use the whole data set to select features correlated, then they will also be partially from test set, right?
No. No, because I understand what you're saying, but no, because I didn't use all of the features. I didn't use all of the samples, let's say, in that part. Okay, so yeah, the features were selected, like all the features were there, but not all the samples were there. So it was split 80-20.
Oh.
Does that make sense?
No, well, maybe I didn't get it. So if you use the whole sample set, you find CPG sites that have the best correlation with osteoporosis, then it means that they will also have good correlation not only in training set, but also in test set. So they will perform already, they have high chances to predict.
So his question is what was training and what was testing,
I
basically,
got you, yeah, so
right?
the correlations
Is
were
that the
only.
right question?
The
Where
que-
was the test? Was that done in testing, training, or the whole sample?
Yeah, the question is features, if features were selected in the whole data set or
Right.
only in training set.
Yeah.
So the 80, when I split it 80-20, that's when I ran the correlations
After that,
after that.
okay.
Okay,
Yeah, makes
yeah.
sense. Sorry, yep. So the first time, what gave me the first look is when I did the weight controlled Bruscal Wallace to look at everything, and that gave me the first round of things to look at, but then the correlations determined what would be retained.
Okay.
How do you measure T-score in the clinic?
So it's like, how do I describe it? It's a
It's like a
scanner.
DEXA. Yeah, you put
Oh, it's a
your
DEXA
foot
scan?
in it. Yeah.
DEXA scan?
Yeah, it's not diagnostic, but it gives you a fairly good indication. So women are about twice as likely to be osteopenic or osteoporotic compared to men. So yeah, it's relatively short. You essentially take your shoe and your sock off. You put your foot into this little scanner, and then it gives you a readout of the T-score.
Did you run your analysis separately by men and women?
Yes, so when we do it that way, we see some less results, but anything that was kept in the. Sorry, no, anything that was kept in the blue here,
Yeah,
they were pretty much retained.
okay.
So maybe one or two weren't, but most were.
Because yeah, I'd be concerned that controlling for sex is probably not enough to get out the
I
effect
agree.
of having a wife.
But it is class imbalance because more females that present with
Yeah.
osteoporosis, so I was concerned about that as well. But then I ran it a second way with regressions to see if it would get the same effect, and it didn't look that pretty, so I wanted to.
Well.
So yeah.
So you're gonna look at the genes, and
Yeah,
do some-
I've looked at the genes already, and they're usually associated with illnesses in children. That's the exciting
Oh,
part.
yeah,
but
you
they're
sorry,
never
you
looked at
presented
aging.
that.
Yeah,
Yeah, okay, sorry.
so
And why is it called OsAge?
because everything else was called age and os is born in Latin, so.
Yeah.
I won't be ready though.
Maybe you don't need the age. you just-
Probably not, well, if I just call it os, like, you know.
It doesn't have much pizzazz.
Yeah, that's fun.
Klopack, talking about utilizing multi-omics and machine learning to investigate general and system-specific
7
aging.
people, you're the only two. Because you can't carry it around.
Yeah.
All right, yeah, very excited to be here to talk to everybody today. So I'm gonna talk about some work we've been doing trying to get at sort of general multi-system aging that the geroscience hypothesis tends to focus on, as well as system-specific aging, sort of within biological system. So I'm assuming everyone here is pretty familiar with the geroscience hypothesis, given the theme here. But the idea is generally aging is this coordinated decline that's happening across systems and across all biological levels. So this is something that's happening over time across your whole system, sort of as one part ages, another part ages, the whole body ages. But there's been some recent evidence that aging is not actually occurring simultaneously across biological systems. So here I've got a little cartoon example showing sort of in green the sort of geroscience, general multi-system aging idea. There's a sort of general aging in the sort of individual person, but they might have a faster cardiovascular aging and a slower renal aging. So for the sort of cartoon imaginary person, they're more likely to have morbidity and mortality risk related to their cardiovascular aging compared to their renal aging. But this sort of difference and this variance across systems is not going to be captured by the ways that we've been trying to capture general multi-system aging, like the epigenetic clocks. So the goals that we had here were to first see if we could describe these sort of system-specific aging scores. And to do this, we're going to use some of the biomarker data available in the health and retirement study. After generating these scores, we then were interested in identifying which biomarkers within these scores have the greatest relative importance for mortality within each system. So basically, is there one or small set of biomarkers within a given system that best characterizes mortality risk? And then finally, to examine which biological systems are of the greatest importance for mortality. So in comparison to sort of a general aging trajectory, is one biological system of greater or lesser import for mortality? To do this, we're going to use the health and retirement study. Again, we're using the data from the 2016 venous blood study. And we took all of the biomarkers that were available and sorted them into 12 biological systems, as well as a 13th general multi-system aging set. And I'll show you what those look like in just a second. To generate our scores of system-specific and general multi-system aging, we used extreme gradient boosting, or XGBoost. So for folks who are familiar with machine learning, this is similar to a random forest. But instead of just a random set of ensembles of trees, each tree is learning from the previous one. And this has kind of been the hot system in, or the hot method in machine learning lately, because it has a lot of utility for highly correlated variables. It can look at some nonlinear associations among variables, which is all very valuable for things like biomarkers where they're very highly correlated with each other. And then we trained these, our criterion variable was 6-year all-cause mortality. So I'm not going to talk too much about the HRS, but it is a nationally representative sample of Americans over the age of 50. The venous blood sample in particular is age 56 and up. There's large numbers of African Americans and Hispanic participants. And there's also a very wealth of biomarkers from both the 2016 venous blood collection, and we also include a few markers from the 2014 and 2016 face-to-face interviews, which includes things like blood pressure. So we have this information on about 4,000 people over the age of 55. So we have our 63 markers. We're going to divide them into physiological systems, and we're going to predict mortality. So here are the systems that I think we're able to characterize in HRS. So we can look at sort of a neurological, nutrition, cardiovascular, endocrine, innate immune, respiratory, adaptive, the sort of general system, circulatory, liver, lipid, metabolic / glycemic, and a sort of renal system. So here then are the actual markers in each of these systems. So you can see we actually do a pretty good job of characterizing most of these. I want to point out that for the endocrine system, we only have one marker of functioning, it's DHEAS. And so in future models, when it's included, it's going to be by itself. I can't train a XGBoost model with one feature. But for all the other ones, these are sort of clustered into these systems, and then each XGBoost model was developed with each of these sets of markers. I also wanted to highlight here the general multi-system aging marker system that is meant to characterize sort of this geroscience hypothesis. So we're using telomere length, mitochondrial DNA copy number, and IGF-1, which are all fairly standard general aging measures. For epigenetic aging, what we did was we actually ran a CFA and saved the scores out of that instead of trying to introduce all of the clocks at once. We also did try all of the clocks at once, and we tried just using one clock at a time. They all kind of behave about the same in that if you put them all in, it picks one. If you put them in as a factor, the factor does about as well as that clock did. If you just do one clock at a time, the factor does about as well as that did. This just seemed like the most sort of clean way of doing it. In terms of other measures, our outcome again is 6-year all-cause mortality. In terms of sociodemographic factors that we're going to use later in the models, we use chronological age, sex/gender, years of education, and race/ethnicity. So that was kind of a lot. So I have now what is hopefully a very entertaining set of swooping shapes that will explain what we actually did in a clear way. So we start with our big pile of 63 biomarkers represented by this sort of big gray box. And we sorted them into biological systems. So I have, I think, 4 colors here. It looks really bad on my screen. It looks a little better up there. In reality, there's 13 sets that I showed earlier, but for the cartoon here, it's only 4. So for each of these systems, we took the biomarkers in each system and used that to train an XGBoost mortality model using 6-year mortality. And each of those XGBoost models allows us to create an algorithm basically comes out of it. So we'll have 13 system-specific aging algorithms that we can get 13 system-specific aging scores out of. XGBoost is a cool model also because it is one of the explainable machine learning models in that it actually can tell us the relative importance of the features, in this case biomarkers, in the model. So we're going to examine the sort of relative importance of the biomarkers within system. Okay, then we're going to move over into our testing data. We did a 50-50 split between training and testing. So in our 50% testing data, we're going to estimate system-specific risk measures using the algorithms defined in the training data. And then we're going to use those to evaluate how well or how important are these systems, each system specific risk for mortality. Yes, it's finally the end of the methods. So exciting part here now, results. So this is from this slide prior in the model. So I'm going to just have to ask you to trust me that I correctly ran the models, and now we're just going to jump straight to looking at the actual importance that came out of them. So here, I'm going to zoom in on this, so don't squint too much right now. I have these come up in much bigger, easier to see ways. But what this is then is the darkness of each of these squares represents an importance value relative to the other markers in that system. And then I multiplied those by the R squared for that system. So if a system did not do very much in explaining mortality, it's not going to have any dark squares. So zooming in here, starting with that sort of general multi-system aging set, we see that telomere length and epigenetic aging come up as the biggest factors in predicting mortality. Sliding down here to adaptive immune aging. Oh wow, this looks like it's not even showing up on my screen. It's a little darker, but yeah, you can see CD8 naive cells show up as the most important biomarker for mortality among these adaptive immune cells. In terms of cardiovascular risk, we get this sort of runaway success for NT-proBNP. This is a marker that's often used to evaluate heart failure, so it is a big indicator that something's wrong with your cardiovascular system. So it's not surprising that it pops up here. In terms of the hepatic system, transaminases, which is ALT and AST added together. Again, these are kind of the go-to clinical markers you would use to see if you are in liver failure. In terms of the innate immune system, we get a little bit of signal at TNFR1, but GDF15 really, again, is kind of a runaway success here. And this is also kind of consistent with some recent research showing GDF15 is a really great just aging biomarker in general, but urine specifically among the innate immune markers, it's quite strong. Down here among the neurological biomarkers, again, not a big surprise, but NFL is again the strongest by far of these predictors. And that's kind of what we've been seeing is among the neurological markers, we have NFL, at least in blood, is one of the better of those markers. In terms of renal, eGFR, which is again a clinical measure of how well your kidneys are doing, shows up quite strong. And then finally, in terms of respiratory functioning, peak expiratory flow, which is also a fairly expected marker of lung functioning. Okay, so that all came from the training data. So we trained all of those systems and then we used those algorithms that were developed in the training set and we put them into the testing set, which is again 50-50 split, so about 2,000 people. And in this testing set, we did another XGBoost model to see which of these systems is most important for mortality. So starting here, you can see that the strongest one, oh, I think actually I have something that pops up. Nope, I don't. So, so this is what it looks like here without any sort of additional controls. We additionally wanted to control for things like chronological age, sex, gender, race, ethnicity, and years of education. So you can see there's a little bit of attenuation in general when you're adding controls, and that's mainly coming from chronological age, which isn't, super surprising. But in general, the systems that are most important whether or not you're adding these sort of socio-demographic factors into the model. And those systems are going to be general multi-system aging. So this is nice because the geroscience hypothesis does seem to be picking up on something, even if you're trying to tease out very specifically this system-specific effect. There is still a general effect that seems to be important here. The cardiovascular system appears to have a quite strong effect, as well as the innate immune, neurological, renal, and respiratory systems. And then, of course, here, chronological age, not surprisingly, is also a strong predictor of mortality, even including all of these biological systems. So a number of limitations here. This is an all US sample of older adults. There's going to be different patterns of mortality across countries. Things would almost certainly be different in different age groups. One of the next steps we're working on is just separating the HRS into the older HRS and the younger HRS and seeing what the differences are between those groups. Blood data were only available at one time point, so we can't establish temporal ordering here. So we don't know how changes in exposures or interventions might affect general or system-specific aging yet. So many large studies like HRS are collecting or have recently collected longitudinal biological data. This is not going to be likely very useful to other studies as a score in the same way that epigenetic clocks are, because no one else has this exact same set of a very large number of biomarkers. But I think one of the things that this does is it shows that there's kind of a smaller set of biomarkers that might be useful and cheaper if you want to go on and just assay those, that actually get at a lot of the mortality effect here. So here, again, within systems, there's often one or two markers. So for adaptive immune, CD8 naive cells are really doing a lot of the work. For general aging, it appears to be telomere length, and epigenetic aging, cardiovascular, NT-proBNP, the hepatic system, transaminases, the innate immune system, GDF15, neurological NFL, renal eGFR, respiratory peak expiratory flow. So if you're going to go out and get a smaller set of markers, these might be good ones. I don't know how much of a money-saving this will really be, because if you do a general panel, you get a large number of these that aren't in this list. But if you really wanted to go out and get a couple extra things in addition to your general panel, things like GDF15 and NFL look like they're really having a strong effect, at least in HRS. And then again, several systems have large associations with mortality, including that general multi-system geroscience hypothesis system, but as well cardiovascular system, innate immune, neurological, renal, and respiratory. Yeah, that's all. Thank you. Wanted to point out. Thank you.
Thanks, Eric. That was really interesting. This is something I haven't thought about before, but I'm curious if it would just enhance the precision of your results. Why not eliminate people that died from accidental causes?
The reason I don't do that is because I don't know who died of accidental causes.
You know, in the HRS you do, right?
It's protected data
Yeah.
that we don't.
I thought, well, I've used like is it in the exit files?
So not everyone has then that information from the exit file.
I mean, you could.
It's with
I think that
yeah.
a lot of people, I mean, because that's like almost 10% of the population and or it would be interesting to look at different causes of death, you know, from metabolic
That is actually also.
from, you know, and see if some of these predict
Yeah.
more within those types of deaths.
So what I've done so far with I think only 4-year mortality we actually have cause of death on also. Because I've looked at least that cardiovascular system and the cardiovascular and a few other things set. And you are more likely to die of cardiovascular disease if you have a higher score on that cardiovascular system age.
Okay.
If you put that and general aging in the model at the same time, it's like 0.06. So I think maybe with additional power in the 6-year mortality, when we get more data,
Yeah.
that might come up. But there's at least some marginal evidence that if your cardiovascular system is more accelerated, even including in the model your general aging, you're going to have more likely to die of a cardiovascular event. So that's all very suggestive, and I don't show anything because.
Yeah, that would be interesting, yeah.
Right, yeah, but that is definitely next steps.
Yeah.
That was great work, and I think you and Rob are having a fight for the most glitzy presentation as well. Well, it's gonna. So you mentioned the UCFA. What informed your CFA? Because the CFA is usually informed by an exploratory factor analysis first, right? Or did you just say,
No.
this is what I think these systems cluster into, so I'm gonna define my factor?
Right,
with-
it is just actually 3 clocks, so it is just pheno age, grem age, and
So-
pace. And it's
it's
partly
only-
because
It's only
the
the-
first gen clocks just do not hang in the same
Sorry, I
factor.
thought you'd done the CFA for each of your
No,
system
it was
components.
just for the epigenetic clocks.
Okay.
Yeah.
And then one of the other things, it's not specific to you, I think how we do this generally
Mhm.
is how much of a confound is something like medications when you're doing this type of machine learning approaches? Because, you know, I didn't get, you had so many biomarkers.
Yeah.
Was the lipid panels included in there because they didn't come out
Mhm.
on the other side? And some of the ones that M had selected, like pulse pressure, didn't
Yeah.
seem to be in there, but NT-proBNP was.
Yes.
Yeah.
Yeah, so and then I wonder about, is that because
Yeah.
we
We're
medicate-
controlling those.
Yeah, we're controlling them away
Yeah,
in the analysis. That's a general.
I think that's absolutely right, yeah. And also transaminases, I'm surprised they show up at all, because it's another thing you screen for and try to really intervene on. The things, that we actually screen for and a lot don't come up here, I think, because we're screening for them.
And treat
So I
it.
think you're absolutely right, yeah. Yeah.
Great talk. Did you see any interaction between these subsystems? Did we
So,
cluster
yeah, I didn't do any interaction analysis. This was just letting the machine, interactions are included, but they're sort of in the black box part of the machine learning.
But I mean, if you look at predictions for every of your subsystems, do you see that some of them cluster together?
Oh.
Maybe, for example, lipid panel and cardiovascular panel.
Yeah, I haven't looked at that, but that is a good idea. I'll look at that. That is a.
Because then maybe you
Yeah,
will
yeah.
see that some of them can be combined because we're
Right.
more or less the same thing.
Yes, yeah.
And you can even, reduce the number.
I will say to some extent, the results of that machine learning model, including all of the systems, pretty much replicates the area under the curve of each of the predictive models, if that makes sense. So it's kind of it doesn't look very different from if you just looked at them one at a time. So they're not, doesn't look like they're really competing a lot for the same variance in that way.
Great, thank you.
Yeah, thank you.
This is really good. The thing I kept thinking of right at the end when you're giving the list of things that were selected was how much that looked like 2018 Justice Tame assays paper, right? So like we use that for other studies to say, here, if we can only afford 5 assays, 10
Right.
assays, this is the set.
Mhm.
It overlapped, other than NFL.
Yeah.
It overlapped incredibly.
Yeah, so I think that's why we added GDF 15 in the
Yes,
first
it is.
place, right?
That
Yeah.
was, exactly. That was the
Yeah,
one we were
so I don't know,
missing.
at one point I was like, oh, someone should just create the tame version of this
Mhm.
compared to this, and then like, maybe they're kind of interchangeable.
Yeah.
Then you would have kind of an externally driven
Mhm.
clock or score of some sort, and then you have just more. Does that make sense? Because I mean, that's what you always worry about when you're creating something,
Right.
is that you're overfitting somehow.
Yeah.
And in some ways, they gave you the list, and that list looks exactly like, pretty much like your list, other than NFL.
Yeah, Yeah, I think that's right.
Which hadn't been.
Which hadn't been, which
In
wasn't,
the
wasn't a
literature, yeah.
possibility.
Exactly,
Yeah,
yeah.
yeah, that's my point, yeah.
Yeah, that's a great point. I'll look at that.
That's a good point.
Thank you, everybody.
So this is definitely an ugly
Yeah,
version.
it looks really well.
Of the parasites, I think. So this work that I'm presenting on today is basically using the same machine learning approach in terms of actually boost and thinking about how this might identify differences in association between blood-based biomarkers and later life health. And I'll explain what I mean by that across race of ethnicity. And this is Klopack, who has taught me quite a bit actually about boost, and I did some reading afterwards, and Leigh Crimmins, who have been very skeptical. We kind of improved this as it's gone through the process. So this is under it's being revised and it's gonna go back under review in like a week or two, so I would welcome questions. But before I begin, the thing I forgot to kind of include in this slide, just because I kind of assumed was actually what I mean by race ethnicity. I just want to be very, very clear. It's a social construct, but not only is it a social construct, it's a social construct with many different types of kind of consequences to when we think about life course exposures and what that means for the aging process, right? So I am not thinking about ancestry, I'm not engaging in that in here. I'm really thinking about the ways in which race ethnicity in the United States is kind of related to the ways of social patterning of both exposures and then what those exposures might mean. So when we think about sort of geroscience advancements and why I think this kind of work is important from a sort of perspective or exploratory manners, geroscience has had a really big development and creation of predictive measures of aging, right? We can think about the epigenetic clocks, we can think about these machine learning approaches where we're really kind of pooling the best biomarkers and we're started trying to predict an outcome and explore that elsewhere. But really less is understood of how these predictive biomarkers may be associated with health and aging differently across race ethnicity. So while I think we had a lot of advancements in terms of that prediction and in terms of thinking about the biomarkers that we should be collecting, there's been less attention in terms of thinking about, okay, we collect these markers, now are they actually predicting the same? And if they're not, we need to also kind of think about the ways in which we're designing our research questions, right? And then racially minoritized populations may have differential associations due to differences in health exposures and resources, right? So the idea here is thinking that, especially for non-white population in the United States, there's different occupational structures, different early life environments that really might be impacting the way specific types of aging are occurring and sort of the process, rather than sort of global understanding of it. So potential differences are we're really thinking about the social patterning of exposures in terms of what people are exposed to across their life. This might lead to different signals of aging or different biomarkers that might go up or down. So when we're thinking about the biomarkers, it's not just only the levels, so it could be differences in CRP, there's also the associations of those biomarkers with those health outcomes might also differ. And then a large part of what this research was kind of interested in is thinking about how predicted biomarkers of aging might actually be tailored towards white older adults, since those are the majorities of our samples, even when we have more diverse samples like the HRS, especially if we don't kind of consider that into the sort of design in the way that we're creating these either surrogate measures or in the ways that we're selecting these biomarkers over time. And then a third question here, and this actually came from an earlier study that I did with academia where we're thinking about biological aging and the 22 markers, and sort of unexpected patterns came up in terms of thinking of Latinos. But I kept thinking about what is it in terms of general versus system specific and how those might differ. And I would argue that for race ethnic differences, for things like all-cause mortality, which I'm preemptively going to talk about in a second, which is really important for population-based surveillance, I imagine that the biomarkers being selected might be very different versus something that's more system specific or pathogenically defined like diabetes. I imagine the biomarkers will actually be relatively similar across those groups. And here I'm thinking about sort of the inputs and the ways in which the exposures across our life are shaping, right, sort of our aging processes or multiple processes like Eric was alluding to. So for general outcomes, this could be disability, multi-morbidity, all-cause mortality. In this research, we don't look at disability, we're looking at multi-morbidity and all-cause mortality. That might have multiple pathologies and risk. People die for multiple different reasons, right? And that can be also be age structured. And then for system-specific outcomes, we can really think about diabetes and heart conditions, which are the two that I also pulled from. And that can be from a narrow set of sort of pathologies and factors that we know are very much associated with those outcomes, like HbA1c, hypertension, NT-pro. I can never say that one. NT-pro, sorry. So the research question is very it was very direct, I think. So the question is saying, do decision trees select different sets of biomarkers for Black, Hispanic, and white older adults for general and system-specific aging outcomes? And here we're using the HRS, which is the venous blood sample, right, 56 plus, multiple biomarkers, completed 2 months within the interview, and there's a 50-50 split, same thing that we've done before. We're also going to do race stratified analyses. So the health outcomes were mortality, so yes, no, that this happened from 2016 after, right, blood was collected to the early part of the 2021, 2020. Multimorbidity, which was basically a doctor diagnosis of all these conditions for hypertension, diabetes, cancer, lung disease, heart disease, stroke, psychiatric problems, and arthritis. I think we actually took psychiatric problems out. Sorry, I did it both ways, so it's actually 0.7. And then diabetes, yes, no. And then heart conditions, whether or not you were doctor diagnosed with any of these types of heart conditions. The biomarkers, I mean, Eric showed them to you a little bit earlier. I have a slightly smaller subset because I wasn't looking at the epigenetics or the cognitive aging, so those were not included in here. But there are quite a few as we go through, and then each of these will also have age and sex gender, which are not biomarkers, but I want to actually at least indicate that those were going to be in the features that we predicted from the model. So analytical approach, and Eric kind of alluded to this already, so I don't really need to go into great detail, but it's XGBoost, the gradient decision trees, right, which is really helpful in thinking about efficiency and accuracy because it's based off prior trees and it's improving through the residuals. And there's regularization and flexibility in it. And then the really important factor, I think, for us is thinking about sort of feature importance and interpretability. So we can look at which are the biomarkers that are being selected and how important are those biomarkers, right, for the outcome of interest. So the first set of results, and I also, if you would like, I didn't focus on the descriptive table that we have. We do have one available. There's a whole paper. Please send me an email afterwards if you want to take a look at it. But for all-cause mortality, and what was kind of striking here is that there's actually only 3 biomarkers out of the top 15, which I'm not showing you the top 15 here. I'm showing you the ones that are most important. Only 3 biomarkers overlap between all 3 groups, right? So this is NT-proBNP, age, and exposure peak flow, and we can kind of talk about why that is, especially in the Q&A. I think those kind of make sense. But what's really interesting, if you look at the top one for Black older adults, albumin, those don't even register for Hispanics. And white, in fact, the level was actually very, very small. Like it didn't even make it to top 15 for those 2. And then you just see some sort of variation between those groups in terms of like chloride level, TNF receptors, and things like that. So next, if you look at multi-morbidity, it starts to kind of come together a little bit closer, which kind of makes sense to us since, right, there's specific conditions that we're looking at. And there's more overlap, slightly differences in the importance across those. And then the last 2, which make complete sense to us when we're thinking about diabetes, it's HbA1c, glucose are the best predictors. This is really much, right, defined also by this partly. And then heart conditions with NT-proBNP was the big one that kind of struck out. And then there were some differences in the importance across those. So that was actually really fast. It was the last lecture of the day, but I can also send you the paper. But the important part to note here is that there are variations in the general, right? And then not so much when we get into the system specific. So, for discussion, which I think was kind of helping us think through this, is that biological mechanisms and risks may vary across race and ethnicity, right? And these can be due to differences in the experiences. I think we should incorporate this more in the type of work that we do. This is not something that this project is able to kind of examine explicitly, but that's something that when we're designing sort of our studies and we're thinking about including these general age markers, we should really be thinking about, does this mean the same, and why it might actually differ across some of those groups. And these things are socially patterned. There are potential limitations in understanding health for racially and ethnically minoritized populations and health inequalities more broadly if we continue just to use these general markers that were trained and predictive for health among white older adults. And then when we're designing these, we should really be thinking about considerations for system-specific and general aging processes as well at the same time. So that was very short, but thank you to my collaborators, also an undergraduate assistant, and then a bunch of funding. And I'll sit here for questions, but thanks to Eric, I didn't have to go into XP Boost or any of that.
So in a lot of these model building things, there's this competition process that can be actually, sort of very brittle with small sampling variability where you've got two competing predictors, one gets knocked off and the other one owns the show in this subset, and then due to almost pure random sampling. So in some, XG Boost is actually nice in general in handling that kind of stuff, but I think most of us just don't in our bones feel like I can totally trust this thing, to generate these kinds of qualitative conclusions quite yet. So have you been able to go back and just apply a standard OLS linear model and find a statistically significant interaction term that says, this slope of association really is systematically different in, let's say, Hispanics versus, non-Hispanic Blacks for this particular predictor? I would imagine at some point, some old reviewer who doesn't trust this newfangled new stuff is going to say, show it to me the old-fashioned statistical interaction way.
So we haven't done that yet. I think that is a sort of next step to kind of go along. The one thing I will say though is also we did bootstrapping for this, and the 95% confidence interval was actually
so
they're predicting the same.
And so have you actually computed though the difference between there's I guess in the model selection there is no coefficient for.
So so there's an importance. I wouldn't call it it's weird.
That's the key thing. Is importance actually isn't the same thing as
exactly. So that's a big limitation I think for this.
It's not really a limitation as long as you can just say that in fact there is a qualitatively statistically significant difference in, let's say, the association between naive B cell percentage and our condition outcomes for Blacks versus Hispanics or Hispanics versus whites. It's not a big deal, but I just imagine importance is always going to beat people out.
Can I ask, just a follow-up to that, which was, so, if we were thinking about the classic statistical way, you would be like, oh, well, within HRS, white population is a lot larger, so therefore you'll see more things. How does that relationship of sample size affect some of these coefficients? Because I was like multimorbidity is what I was expecting to see across all these conditions. It's like everything had an important score for whites, and then it was different for Black and Hispanic, but the mortality was actually completely reversed, where there were many fewer things for white. So I don't know, my intuition is failing here. What should I be thinking about with sample size and importance scores?
To be honest, I don't know exactly how to answer that question.
That's a great.
Yeah, and I think this is really a first pass. There definitely was a sample size concern. I will say, at least also when we think about the events and what's powering this, at least all these were above 100 events that were kind of occurring. And the other part too is which I would like to do eventually, thinking from a demographic perspective, is thinking at the different ages too, or different clusters. 65 to 75 is going to look very different from 75 to 12.
Right.
But yeah, no, we are definitely limited.
And the other thing I was wondering if you thought about is mortality selection and the fact that especially, fortunately, individuals from minority backgrounds tend to die earlier sometimes, or so that we might be seeing the healthiest Black people in the HRS and Hispanics. Do you have a sense of how that might affect this, or?
That's where I would go back to sort of the aging and get more longer term, at least because there are the younger 56-year-olds in here that we could kind of
those that don't even make it to 56, you know, yeah. I guess, yeah. You could maybe try to like do something to account for mortality selection and see if that changes your estimates or.
Even the differential age importance scores kind of goes along with what she's saying.
Yeah. Yeah.
We're having an Amber Alert. We ignore it. It's not
Yeah,
a
it's
like
all-
it's just a child abduction.
We're in the second.
We don't.
Get out, hang
The
on a
idea is
tree.
if we're driving on the freeway. Find this,
Quietly walked away. He's like, it's over, I'm out of here.
We're done. Does anyone have a conclusion, or an update, or where we go next? Are you all tired? People have talked a lot. I mean, we have a lot of things to talk about in small groups.
Yeah.
Pretty general.
Can I, it's
say no.
something, I mean, so across lots of talks that I've seen, and it comes back to this point about where we are in terms of the generations of clocks, right? Like, I don't think any of us really got past generation 2. Like, and even with the stuff you're doing, Eric, I think that's got real promise, doing the machine learning. But again, it seems that there's about 8 biomarkers
Right.
that came out, and there's 8 biomarkers in M's clock, there's 8 biomarkers in the Grimmie's clock, and it seems like with any of these, when you've got
Something.
yeah, but you know, a handful of biomarkers, what we know that are associated with aging, you can build a good methylation clock off it, right?
Yeah.
But it doesn't necessarily really inform us about what's causing aging, right?
Mhm.
So there's kind of circularity around all of that stuff, and I don't know how we get beyond it outside of the time dimension, where we can actually maybe start to pull apart the DNA methylation change in response to the biomarkers changing, or vice versa, and or some other third vector. Because occasionally I do wonder, like, are we doing much better than counting the lines on your face, or how grey your hair is, or gum erosion, or you know, because there's algorithms out there to do facial aging and that sort of thing, and I do have a concern as someone who subscribes to a lot of this about where we go. Yeah, where we go.
Well, at least we're not buying things that aren't.
True, we're not selling
perhaps
anything.
Not taking us anywhere. We're being skeptical
Yeah.
and we're being thoughtful about what they are. So,
Also, I think what you said is exactly right. We need longitudinal data, and I think maybe working with people like Jim Kirkland on intervention data.
Not doing more.
What is metformin affecting? What are the senolytics? What are interventions affecting?
And do we do that though, Eric, at the level, are we, so is this like, so for example, in Tilda, we will have 3 sweeps of methylation. Or we've already collected bloods, we just haven't asked the third time point yet for a large number of people. But we have changed in medications over time. How much people really change things like smoking and so on, I don't know. I haven't actually looked at this really across the 6 waves of Tilda data. But yeah, like I do think that's a way to. And are we doing it at the level of the clock, or are we doing it at the level of the CPG sites?
Because you'll know who got on like statins, and you'll see if their lipids went down. You'll see if their lipids going down affect their epigenetic age. So I think you are really in a position with your already having some longitudinal data.
And we do have the because we collect medications histories from people. So when we arrive, when the interviewer goes to the household, we often go off and retrieve all the medicinal packaging that they have, and then we ETC code it. So, but we do that at every wave, so it's not just the health assessment wave. So yeah, we would actually have a good bit of,
very detailed.
Yeah, of that. Now we don't get dose.
Right.
That's a limitation.
That's still more than a lot of people are gonna have.
Yeah, how much does it vary?
Because I think that would be interesting, because occasionally I see these studies appear and it's like in 10 healthy men, and you know that trial where they give them what was it, recombinant growth hormone or something like this here, metformin and something else, and it looked like there was epigenetic age reversion over a 6 month or 1 year
Mhm.
period, and then they kind of go, yeah, how representative that is really, what's really going on, like.
All right,
Stop with my cynicism.
I think people are done, tired. Thank you.